Assalamu alaikum. Good morning. First of all, I want to thank the organizers, especially Dr. Najla Abdullah, for inviting me to be in Cairo. This first time for me here in Egypt. So before I start, I want to show you our campus. This is for pointer. So this is our campus, main campus in Hanover. And so Hanover, we call it also the city of universities. So far there are 5 universities in Hanover, and Leibniz University of Hanover is one of these. And it was founded in 1831, and just a few years ago, the logo of the university was changed and give this a new logo, and the name of Leibniz came from a famous scientist from Hanover. So he implemented this binary system, which everyone using this in computer science, let's say. Also, I will give 2 slides to introduce our institute. So our group of interest mainly legumes. So we are working on legume transformation, mainly different types of legumes. Also, we do some other work on apple, tomato, tobacco, or arachidoses and roses. For field of activities, we are mainly interested in biotic and abiotic stresses, like fungi, insect, virus, drought, tolerance. We did some work on molecular farming, and we also interested in expression stability and expression analysis. We did also gene stacking, so now we have pea plants containing 4 different antifungal genes. Also, we are interested in RNAi work. So here are examples of the 4 genes which we are working mainly. So catenases from Streptomyces, BST from grape, BJ from kiwi, Leucanase from barley. As I told you, now we have clones containing this all 4 genes. Unfortunately, we couldn't test these plants in Hanover, and we had to send these seeds to Canada to make field trial there. Now I will come to the topic of my presentation, so development of insect-resistant transgenic pea. This was master work of one our student, Nigawa from Ethiopia, so I will try to present his result. This will be the outline of my presentation, brief introduction, material method, result, discussion, and then the summary. As you know, legumes and pea are one of the main staple foods all over the world due to its importance as food and feed. It contains around 40% of protein, very good amino acid profile. It has very nice ability to make nitrogen fixation, and this reduce application of nitrogen fertilizer by 40%. Pea can be used as model plant for more research also. If we come to pea production, we can see here the countries which are producing pea. If we go to regions, we can also see the difference between African and Asian countries and European or North American countries. So there are gross difference in the production. If we come to the country itself, also we can see there are a lot of difference between different countries, and this can be contributed to different production constraints, abiotic or biotic constraints. And today I will talk only about insects, which are responsible for up to 40% yield losses. And here are a few examples of most important pests in pea, like pea weevil, pea moth, pea leaf weevil, and aphids. There are, as you heard from different presentations, there are different methods or systems to overcome or control this pest, either using resistant varieties, but the drawback of this that pest or insect can overcome the resistance in a few years, or using chemicals, and this, of course, it will damage the environment and also the human health. As you can see, this guy is spraying without any mask or without any protection. We can use political methods or different cultural or regulatory methods like certified seeds or different methods. And of course, the most important one, let's say from this conference, that the technology tools using BT or CRY genes. So BT is gram-positive bacteria. It was identified or known in 1900, and then it was called Bacillus thuringiensis in 1930s after German scientist Berliner. And thuringians are coming from a German state, Thuringen. It's in former Germany. And this bacteria produce crystalline proteins, and these proteins were used widely in commercial agriculture and also organic agriculture, as you can see here. So before it was no problem to use these spores for spraying, but later when it came to transgenic, it became more problem. So the mode of action that the protoxin should be activated, and this done normally in the gut of insect due to alkaline pH and protease. Then the active protein will recognize the receptor, bind to receptor, and after that it will make some pores or holes in the gut of the insect, leading to that crystals and the bacteria which are in the gut of insect, it will go inside and proliferate and lead to lysis of the cell. So the insect eat the spores, let's say, then after recognizing the activation of the protein and bind to the receptor, the insect stop eating, and later so the insect will die. So this is the action. Regarding P, we are interested in two types, let's say, either for field-based, and this mainly P mold, African bollworm, or tobacco bollworm. And for this, we can use CRY1AC or CRY1AB, and this targeted to Lepidoptera. We have another problem for the storage of peas, because this also make around 100% of destroy of the yield in the storage, and the quality, of course, and also mycotoxin will do different problem. And this, we can use a CRY3 group, let's say, was targeted for Coleoptera. And the main problem for P is P weevil. Regarding P transformation, there are different protocols or genes used for transformation, early using some marker genes or herbicide-resistant genes or antifungal genes. This from our group, and or chitinase gene from Streptomyces. But for CRY gene, we think this first time someone did this work for P. This is the vector which we used, and we are using B green system, and with the helper plasmid called B soup. We are using Agrobacterium strain EHA105. In the vector, you can see as selectable marker, we are using BAR gene, which confers resistance to herbicide, W35S promoter, and then CRY-AB or CRY-AC. Transformation step itself starts from preparation of the seed, then embryo access, inoculation with Agrobacterium, co-cultivation, washing steps, then regeneration and selectable steps, until we get quotative transgenic plants to go for rotting. And as Soumya mentioned yesterday or before yesterday for chickpea transformation, we're also doing in vitro grafting. Either we use this seance grafted directly in vitro grown seedling, or we germinate the white type seed in soil or black soil. Then after grafting this, we wait until we get the T0 seeds. For molecular characterization, of course, we go through all these steps from DNA, RNA, protein, tribes, using different systems, PCR, savan blot, transcription, and translation analysis. We're using different methods which I will show you briefly. For feeding test, we use tobacco bollworm, and here the life cycle of this insect, this adult, lay eggs, develop to larva, and then bollworm, and then this is the life cycle. For the experiment itself, we use two different types. detached leaf feeding, and contained whole plants. The idea to know the larval survival and the feeding damage on the leaves. In contained whole plants, we were interested in hatching percentage, larval survival, and damage on the whole plant. Now I will show you the result. First of all, leaf paint. So normally we apply the herbicide solution in one leaflet of the plant. The other one is control. And after 1 week, we can see the result of leaf paint. If the applied or treated leaf is healthy, we can call it bar positive. If there are partial necrotic, we can say partial bar positive. Sometimes it's negative, so there are no expression for bar gene, it's bar negative. And you can compare it with negative control, so the treated leaflet is totally necrotic and died. At PCR level, we are using multiplex PCR using two set of primers, HMG for control gene and Cry1 AC primers, and all this T0 clone show integration and the presence of the genes of interest. In the lower figure, we use bar gene to confirm that bar gene also integrated in the genome of plant. We use Southern blot to study the copy number and integration better. Some clones give single copy, some give multiple copies. I think the photo maybe is not so clear, but at least you can see here there's two single copies, there's multiple copies, transgenic clones. For segregation analysis, we use PCR, again using these two set of primers. And you can see here at the two generation of progenies, some of them have, of course, it is transgenic, some are only one band, so we select this. We went to T3 generation again, so we make all this PCRs to see the segregation and perform homozygosity. This is the result of T4, and these are coming from the red or blue plants, as you can see here. So most of these clones are homozygous now. And some of this blue box, some of these plants still segregating, and there are different percentage of homozygosity. And so we have to go for further generation until we get really 100% homozygous line. At the transcription level, we did, of course, RT-PCR. Again, it's a clear result that Cry gene is transcribed, and we can see the difference between this negative control, positive controls, and there are genomic DNA, so there are no contamination because the HMG gene, which we use for control PCR, is contained in tron, so there are shifts in the band size from this 570 to 350, which tell us that we have a clean RNA preparation. In order to study the exhibition of Cry-AC proteins, we use this lateral strips or amino strips, and easily after preparation of the total protein from leaves, we put the strips, and then we can see the test line, which tells that it contains Cry-A protein or not. The control line, it will tell that the work is fine, but the important one is test file. All these clones showing exhibition of a Cry gene, and you can compare to the first line here. This is negative control, which are missing this line. If you come to feeding test, in the first experiment we use detached leaf, and here in this you can see the larval survival. So there are clear difference between control and transgenic clones. And if we come to how many days need to reduce the larva, you can see after 3 days, even the control, the larva and the control also drop to 40%. The point here that the leaves after a few days become dry, and so it also clear that even the larva and control plants are also not survived. And here the visual scene between transgenic plants and control plants. For that we thought to use the whole plant, and for this we lay eggs on the plant, this non-transgenic, transgenic, so there are different eggs in the plants. And to, first of all, to see the hatching percentage of the eggs. And yeah, of course, it should not be too much difference between control and transgenic plants because there's nothing to do with the cry gene at this step. But if we go to the mortality or larval survival, you can see even in the transgenic plant there are 55% of eggs hatched, but the important thing that 0% survived. In the control plant, there are around 86% hatched eggs, and more than 90% were surviving on the control plant. So this is an important finding, let's say. And here you can see the difference between transgenic plant and non-transgenic plant. There are different. The effect of feeding on the leaves, you can see, while in transgenic plant there are only where the eggs were laid, so there's no great damage. For larval survival, of course, it was 0% for transgenic plants and more than 90% for the non-transgenic or control plant. If we look for how many days needed to get the reduction in larva survival, you can see after 2 days in transgenic plants it is around 20%, and after 5 days it's 0%. If you compare with the negative control, that it's reducing, but it's still above 8% after 5 days. And again, here you can see clearly the difference between transgenic plants, which are growing happily and very nicely, and these control plants, which are already dead or eaten, as you can see in these figures. So if I can summarize or to say about the discussion, that larva survival was, or larva mortality was in agreement with other research that it's 100% reduction using also Cry-AC in transgenic rice or tobacco. Also, feeding damage was much reduced in transgenic plants compared to non-transgenic plants. In summary, you can say that we are already produced transgenic pea plants containing Cry-AC genes, and these shoots or plants were grafted successfully. We did all these characterization methods to prove that it's really the gene of interest are integrated and also it's inherited to next generation. Now we have T4 generation and we are growing T5 generation. We still have to make feeding tests to see really if there are any side effect, let's say. In summary, we already developed these lines. We will go for, I think, next step will be crossing of these lines with other lines, so maybe antifungal genes containing lines, so we have more genes in one line. We need also to make some quantification analysis for protein. We have to make some studies also on the non-target organisms like rhizobium. And very important things to make field trials. Unfortunately, this we cannot do in Germany, and we have to look for another country who can accept our plants to be tested there. As I told you, we sent some seeds already to Canada for field trial. Here are the reference, and thank you very much for your